a framework to understand the global evolutionary ecology of urban plants and butterflies. In this study, we asked ourselves this question: how do we create a globally accessible framework for evaluating functional trait variation in urban butterflies and flowering plants? For this study, we used common butterfly cabbage white and herbaceous flowering plant daisy fleabane. With the development of this framework, me and other members of the Pfeiffer-Rixey lab underwent the repetitive process of collecting our sample species, preserving them, analyzing and measuring functional traits, as well as refining the protocol. Starting us off is our butterflies. Collecting our butterflies, we prepared a mason jar consisting of cotton balls slightly soaked in acetone. Alternatively, we also use kill jars, which is a jar pre-made for collecting insects. After catching our specimen in the net, we gently pinched the wings of the butterfly outside the net and carefully inverted the net into the jar. Upon returning to the lab, the butterfly is placed in a small bag and put in the freezer for 24 hours. Following the 24 hours, the butterfly is weighed. After the weight is recorded, the butterfly is pinned. Resorting to various types of materials, we tried dry erasers, staples, paper clips, styrofoam, and sewing pins. After pinning the butterfly, we measure the butterfly's wingspan, the length and width of the forewings and hindwings, as well as the length of the full body. Next is our plants. First, we measure the whole plant from the ground to the tallest point. After recording the measurements, we collected 5 flowers and 3 top leaves as well as 3 bottom leaves from the plant. Upon returning to the lab, we took photo images of the collected flowers and leaves next to a color scale labeled by site. We then pressed the flowers and leaves, leaving them to dry out for a minimum of 72 hours. After the 72 hours, we measured the width and length of leaves, as well as the diameter of the total flower and its pistil. We took the following measurements using a ruler, followed by the ImageJ software. Like mentioned earlier, there were various changes and a lot of refining of the protocol. Here are a few changes we made. As we refined butterfly capturing methods, placing the jar inside the net with the butterfly and cutting off any exits was easier than our original method. The new method minimized any possible damaging of the wing, as well as the chances of the butterfly escaping the net. Upon pinning butterflies, we learned that rehydrating them prior resulted in minimal damage. With further consideration, rehydrating and pinning butterflies was deemed unnecessary due to the purpose of this step being more geared toward displaying butterflies rather than measuring them. After refining the protocol, we determined that plant measurements would be done in the field. In addition, taking images with the color scale would too be done in the field. As a result of taking measurements in the field, pressing the flowers and leaves was not a necessary step.